Hello children, how are you? Fine? So, today we shall be discussing to you the 5th chapter of your NCERT textbook, that is Principles of Inheritance and Variation. Under this topic, we shall be talking to you about Mendel's monohybrid cross. But before we discuss about the monohybrid cross, let us know something which will help you to understand the monohybrid cross. First of all, let us know why the baby elephant look like their parent, or why a mango tree small seedlings when grown, it's looking like the leaves are same like the mature mango tree, or a particular flowering plant, when the seeds are collected, grown somewhere, it will give rise to same type of plant. I mean to say, what is the governing factor which decide which type of flower it will be, which type of plant it will be, which type of fruit it will give rise. This all we talk in this topic, that is 5th chapter of your NCERT textbook, Principles of Inheritance and Variation. Before Mendel, Gregor Johann Mendel, there are a lot of experiment individual scientists done on this direction of how the characters from parents descend down to the offsprings. But for the first time, it was Gregor Johann Mendel who gave a very concise, a very systematic idea in the form of his principles, doing experiment on garden pea. So what was his experiment? Let me first describe, then we shall go to the monohybrid cross. Mendel, he took garden pea plant, for his crossbreeding experiment. Now question comes, why instead of taking other plant he selected pea plant? It is because within a short span of time, a person who is involved in experiment, the Mendel himself, could experiment throughout so many generations to test his observations, to repeat his observations generation after generation before coming to a particular conclusion. So what Mendel did is, he took 7 pairs of contrasting character, one at a time or 2 at a time. What are those? Say number 1, length of plant. Length of plant. Some of the characters I am explaining here. Say in terms of length, either it is tall or dwarf, means short. Tall or dwarf, short. Color of flower. Flower. Either violet or white. Next, color of seed. Color of seed. The seed produced by the pea plant. Either yellow or green. Seed shape of seed. Either round or wrinkled. Round or wrinkled. Likewise, he compared 7 different colors and he tested in his laboratory the crossbreeding with respect to these colors. One by one we shall discuss. So let us discuss on the basis of tall and dwarf first. What he did, tall and dwarf. But before he conducted the crossbreeding experiment between tall and dwarf, he tested the purity, whether the tall is pure line or not. How? By self-pollinating the tall plant for several generations. He tested the purity of dwarf plant, whether it is really dwarf or not. By testing, how he tested? By crossbreeding through several generations. Self-breeding with dwarf plant, considering one male flower, one female flower, self-pollinating through several generations. When after several generations he got that all the plants are short or dwarf, or all the plants are tall, all the plants are short. Here the symbol tall we represent with the help of capital, and the reverse, that is dwarf character, the small alphabet of the same. Is that clear to you? Now, in a diploid cells, we are having a pair of homologous chromosome. During Mendel's time, much of genetics was not known. Anyway, suppose the character governing for tallness, T, a pair of capital T. So one present here, another present on its homologue at the same position called as loci or locus. If this is both are capital T, then it is called as homozygous condition. Homozygous if it is this pair of chromosomes, if the character governing genes small t, dwarf character, then we call small t here homozygous. For the character dwarfness. If it is capital T, small t, suppose one of the gene is mutated. Suppose here in this locus one is capital T, one is small t. What will be the phenotype we shall come later. Then this condition we call heterozygous. Heterozygous. Before we discuss about cross, we have discussed what is homozygous, what is heterozygous, what is the locus position, same position in the homologous pair of chromosome. Why 2 chromosomes? Because of diploid organism. So this terminology you shall have to know before we discuss anything. Now what Mendel did is, during his time, the concept of gene was not there. So he considered he used a terminology that is factor. Factor. And as for his version, each and every character is governed by a pair of factor. This factor today we called as gene, otherwise called allelic gene, present in homologous chromosome at the same locus. So tallness character is governed by Mendelian factor capital T, whereas dwarfness governed by small t, a pair of factor. Now, when a tall plant is crossbreeded with a dwarf plant, what will be the result? This is homozygous tall, a pair of capital T. When they will form gamete, gamete means n number of chromosome will go, means 1 T will go to the gamete, so gamete formation. Here, during gamete formation, 1 T will go to the gamete because they are homozygous, so both are having small t. So either of these two means small t will go to the gamete. Now, when either this is male gamete, this is female gamete, or vice versa, when this gamete fuse to form a zygote, and zygote, that means zygote will be after fertilization and then will produce seed, and seed will give rise to a new plant. What will be the combination? Capital T, small t. That is in F1 generation. F1 means fast filial generation. Is that clear to you? So in F1 generation, the resultant progeny is all are capital T, small t. So what will be the phenotype? So phenotype is tall plant. What is phenotype? Phenotype means the observable character which by observation we can see. So when the seeds of this are grown, they will give a tall plant. That is, we can see the tallness character, the plants are tall. This property we called as phenotype, means observable property which is expressed. Okay? And what is the genotype? Genotype is capital T, small t. Now, when F1 plant, capital T, small t, is crossbreeded with another F1 plant, means when the same F1 plant are taken for breeding practices. So this F1, these are the F1 plant, F1 hybrid. This is F1 hybrid. Let me rub this word. Now, when the F1 hybrid allowed to undergo gamete formation, this one and this one, gamete formation, gamete of F1. F1 means first filial generation. What will be the gamete? As one of the factor, Mendelian factor here, capital T, another is small t, so they will separate from each other. One capital T will reach to one gamete, small t to another gamete. Same as the case for here, capital T to one gamete, because of they are heterozygous, small t to another gamete. So from this If it is a male plant, male flower giving, if it is a female flower, so it is producing two type of male gamete. Female flower producing two type of female gamete. Now, when let me rub this part. When the F1 gametes are fertilizing with each other, random fertilization, this capital T can fertilize this gamete can fertilize this, resulting is capital T, the new generation. It can fertilize this. It's a chance factor. It's a purely chance factor. This capital T can fertilize this small t containing. So capital T, small t, resultant product. This small t can cross with this capital T, resultant is capital T, small t. This small t can cross with this small t, resultant is the small t, small t. So we have got the result. This is called F2 generation. F2. So in F1 generation, we saw F1 hybrid. All are tall plant with the genotype capital T, small t, that is heterozygous tall. Here we have seen what is the phenotype. This is tall, these two are tall, this is dwarf, okay. But do you find any difference? Yes, this is homozygous tall, tall. This is also tall, but it is called heterozygous. The two factors, or today what we call gene or allelic gene, they are capital T and small t, different, heterozygous. Heterozygous tall. And this is dwarf. Both are having same gene or same Mendel, as per Mendel, same factor, small t. So homozygous dwarf. So if we, let me rub this part here, this side. If we see how many tall plants are there in F2 generation, 1, 2, 3. So 3 are tall, 3. How many dwarf plants are there? Only 1 dwarf. Only 1. So the ratio of F2 plant, F2 generation. Ratio between what? Tall is to dwarf is 3:1. 3:1. This is the observable character. We call phenotypic ratio. So let me write in the bracket phenotypic ratio. Phenotypic ratio. But what about the gene character? Gene character is capital T1, pure tall, 1, heterozygous tall, tall, 2, 1 and 1, 2, and homozygous dwarf, that is pure dwarf. dwarf. 1. So the genotypic ratio 1 is to 2 is to 1. That is the genotypic ratio. Genotypic ratio means the ratio of proportion with respect to the gene combination. Why it is here? Gene combination is capital D, here it is capital D, small d, here it is small d, small d. Is that clear to you? So this types of breeding practice we call as monohybrid cross. What is it called? Monohybrid cross. So let me write here monohybrid cross as we are discussing. Why it is called monohybrid cross? How can we define them? Very simple way. When a crossbreeding is done by taking one pair of contrasting character then it is called monohybrid cross. Monohybrid cross. Monohybrid cross. We have taken only one pair of contrasting character. What are the contrasting character? One pair, that is tall and dwarf. Initially we have started with tall crossbreeded with dwarf. So this is tall, this is dwarf. So only one pair. Pair we need to say here tall versus dwarf. Okay. So only one pair of contrasting character we have crossbreed taken into consideration for crossbreeding practices, and this phenomenon we call monohybrid cross. And what is the result of monohybrid cross? Of course, Mendelian monohybrid cross. Result of Mendelian monohybrid cross is, so if I make a summary, let me wrap this part. Result of Mendelian monohybrid cross. What is the result? Number 1, all the F1 plants, this cross we have taken with respect of result of monohybrid Mendelian monohybrid cross with respect to what? Tall and dwarf plant. All F1 plants are tall. What type of tall it is? Heterozygous tall. Heterozygous tall. What about next property? In F2 generation, the phenotypic ratio of tall and dwarf is equal to 3:1. Means for 3 tall plant, 1 dwarf plant. Third, in F2 generation, the genotypic ratio is equal to 1:2:1. Is that very clear to you? I suppose you got a very clear concept. The same monohybrid cross we can do with respect of a round and wrinkle seed, with respect of the violet flower versus white flower or any such types. So from this monohybrid cross, Mendel, let me write this part. So from this monohybrid cross, Mendel came to a conclusion and he deduced, he derived, or rather to say he postulates two principles. One is, so if I rub this part, so from his findings or observations of monohybrid cross, he postulates two principles. One is, which later on we sometime came to know in the form of law, Mendelian laws of inheritance. Mendelian laws of inheritance. What is it? Number one, dominance. Law of dominance. Law of dominance. And law of segregation. You have seen in the Mendelian experiment, what we have seen is when tall plant is crossbreeded with dwarf plant, pure line tall with pure line dwarf, in F1 generation, all the hybrid plant so produce are tall. Although the genetic combination of this hybrid, F1 hybrid, genetic combination of the F1 hybrid is having one capital T, one small t. That means the allelic genes, what you call today gene, Mendelian factor, one is capital T, other is small t. That even then small t resembles for dwarf character, but the expression, phenotypic expression is all the plants are tall. That means somehow the tallness character trying to mask, hide the dwarfness character. That means allele for the tallness property is more powerful here, not able to express the allele of dwarfness. This is what we call, as per Mendelian concept, the law of dominance. Means when a dominant allele is there, recessive allele will not get a chance to express itself in principle dominant allele. That is what we call law of dominance. Is that very clear to you? Well, law of segregation we shall discuss in our next class. So by the time, I suppose you got a very clear concept about monohybrid cross, Mendelian experiment and the outcome, phenotypic ratio, genotypic ratio, genotype and phenotype. For the time being, thank you.